who laugh at an infant who has a heart attack at the age of 2. Let's take a look the pathology. So it is also this hypercholesterolemia is a recessive disorder. Okay, because when the patient has a homozygous recessive allele, then the cell do not express any receptor to absorb LDL. Okay, LDL stands for low density lipoprotein. LDL, low density lipoprotein, also known as bad cholesterol. So in patients with hypercholesterolemia, they do not have the LDL receptor, so that their body cell cannot absorb LDL from the blood into the cell, so that their blood will have very, very high LDL level. But for healthy patient, normal patient with homozygous dominant allele, big H, we have normal expression of LDL receptor. Then when LDL binds to the receptor, that means here, then the receptor can cause something called receptor-mediated endocytosis. So the LDL will be absorbed, will be internalized inside the cell. Then it will lower the blood LDL level. What about heterozygous dominant? Big H, small h. Heterozygous dominant, they have, 50% less of the LDL receptor. So that their blood cholesterol is higher than the normal patient, but their blood cholesterol is high because they only express half of the LDL receptor, so only half of the cholesterol can be absorbed by the cell. The other half of the cholesterol will remain in the blood, causing high blood cholesterol level. That explains why in the homozygous recessive, when there's absolutely no LDL receptor expressed, then the patient will have a very, very high bad cholesterol in the blood, because none of them will be absorbed inside the cell. This is an example of incomplete dominance, because in the heterozygous dominant, in the case of heterozygous dominant, the phenotype, meaning that the expression of the LDL receptor is reduced by half. Let's take a look at another example of the exception. Another example of the exception of Mendelian genetics, we call it codominance. Codominance, the easiest one is to look at the blood type, blood group. Okay, so it involves 3 alleles of a gene. So this gene has 3 alleles instead of 2 alleles, which is interesting. Okay, the dominant allele of the blood group is called IA, and then another, yes, 2 dominant allele. Another dominant allele is called IB. The recessive allele is, small I. So in the next few slides, I'll show you a table. The combination of these 3 alleles, it will give you the 4 blood group. So these 2 are dominant. dominant allele. This one is recessive. So we call it codominant because for determining blood type, we have two dominant alleles. In the next, I'll also explain to you how we what do we mean by blood type A, blood type B, okay? Oh, okay, it shows in here. As you can see that, okay, IA, this is a dominant allele. IB, dominant, is a recessive. I would not say substance, I would say glycoprotein. Glycoprotein, they are like the ID card or passport expressed on the cell surface. So when a red blood cell expresses glycoprotein A, then the red blood cell is the blood type A. When the red blood cell expresses glycoprotein B, then the red blood cell is blood type B. When the red blood cell expresses both glycoprotein A and glycoprotein B, then the red blood cell is blood type AB. When the red blood cell expresses no glycoprotein, then the red blood cell is blood type O. So let's take a look at the, oh, not yet. So it says that there are, okay, we have 3 allele. That means 6 possible genotypes. We'll show you all possible genotypes in the tables. So as I said that IA, IB, they are dominant. Small I is the recessive. So genotype IA, IB, they are the blood type AB. See, type AB blood. We call it codominant because the cell will express both glycoprotein A and glycoprotein B. So in here, when it says carbohydrate, it means glycoprotein on the cell surface. Okay, this is the table I want you to memorize it. Okay, this is the very important table. This slide and the next slide, it will give you a very important table. Just memorize them. So you can see that. Okay, let's take a look at blood type A first. Blood type A, it can have a genotype. This is what we call homozygous dominant, IA, or heterozygous dominant, IA and small I. In either case, it will give you the blood type A. And because on the red blood cell surface you have the glycoprotein A expressed, and then interestingly, in the plasma, that means the blood, so-called the water, the plasma, you have the antibody B. Antibody meaning that it is the protein that will bind to glycoprotein B, or how do I explain this? The antibody B is the protein that will cause aggregation or agglutination of glycoprotein B. So we'll talk about what it means. This is the antibody B, or in a simple term, let's put it this way: antibody B, they can kill the blood type B cell, blood type B, or red blood cell that express glycoprotein B. So okay, the type B blood, the opposite happen. You have homozygous dominant B, and heterozygous dominant B as well. You will express glycoprotein B on the red blood cell surface, and they will have antibody A in the plasma. For blood type AB, you have codominant. Sorry, you have codominant IAIB. This is very interesting. Codominant IAIB. So the cell express both, as you can see that glycoprotein B and glycoprotein A. So it has both glycoprotein expressed, and interestingly the plasma does not have any antibody. Blood type O, the patient will have homozygous recessive allele, small I, and there's no glycoprotein A and no glycoprotein B. And interestingly, the plasma has both antibody A and B. And you also need, ideally you should also memorize the result here. So if you are blood type A, that means the phenotype is A, then what will happen? Blood type A can receive blood, can receive blood transfusion from blood type O and blood type A. But when you receive blood transfusion from blood type B, the red blood cell will agglutinate or aggregate together. Why? Because in the plasma of the blood type A patient, they have antibody B. The antibody B will bind to the blood type B, will bind to the red blood cell B. It will cause agglutination or aggregation, or basically clumping all the red blood cell B together. Bind all the red blood cell B together. And that can kill a patient because all these they form solid in your blood. When this solid goes into your capillary, it will block the capillary. It can cause stroke, it can cause heart attack. That's why wrong blood transfusion can kill. And then blood type A patient also cannot receive blood type AB transfusion. The reason is that on the blood type AB, the red blood cell also has glycoprotein B. Then the antibody B in the plasma of the type A patient will cause the clumping or agglutinate or aggregate of the red blood cell AB together, forming some solid. Similarly, for blood type B, they can receive blood transfusion from O, they can receive blood transfusion from B, but they cannot receive blood transfusion from A, they cannot receive blood transfusion from AB. Blood type AB, they are the luckiest one. They can receive blood transfusion from everyone. Therefore, we call blood type AB universal recipient or universal receiver. So either one is fine, universal recipient or universal receiver. Blood type O is very unlucky because they can only receive blood transfusion from a blood type O. All other blood will cause agglutination. So, but they can donate blood to all other blood type. You see that here. Therefore, we call blood type O universal donor. Universal donor is blood type O. So remember this. So this is the summary of the table. I suggest you to memorize this. Okay, this is important. Print it out or whatever. Okay, let me ask you this question. You need to answer all these questions. Okay, universal donor, universal receiver.